In this topic, we're going to discuss DNA sequencing and gel electrophoresis. So we're going to look at the sequencing of nucleotides of a gene, gel electrophoresis and the different types of gels, the rate of migration of a DNA fragment, reading a DNA sequence from a gel, and then automation of DNA sequencing. Many human diseases have a genetic origin. These are often the result of a gene mutation. Now, recombinant DNA technology has enabled us to diagnose and treat many of these genetic disorders. In doing so, it's often necessary to know the precise sequence of nucleotides of the gene responsible for the disorder. There are a number of different methods to sequence the exact order of nucleotides in a section of DNA. One is called the Sanger method. This uses modified nucleotides that cannot attach the next base in the sequence when they're being joined together. They therefore act as terminators, ending the synthesis of a DNA strand. So in this diagram, the DD represents the terminator nucleotide. So in the first example, you can see that thymine is the terminator nucleotide. So the complementary strands will always end in thymine. The same applies to adenine in the next fragment. So let's have a look at what this method entails. Well, firstly, you need to get multiple copies of the DNA fragment. So you use the polymerase chain reaction, which we discussed in the last topic. The next step involves heating the DNA helix so that it unwinds into single strands. Then you set up 4 different tubes containing the single-stranded fragments of the DNA to be sequenced. So these fragments act as a template for the synthesis of the complementary strands. A mixture of nucleotides with the bases adenine, thymine, guanine, and cytosine is also added. And then a small quantity of one of the 4 terminator nucleotides. For example, in tube 1, you have terminator cytosine, tube 2, terminator guanine, tube 3, terminator adenine, and tube 4, terminator thymine. Then you need DNA primer and DNA polymerase. So DNA primer is used to start the process of DNA synthesis. And this primer is radioactively labeled or labeled with a fluorescent dye. And of course, DNA polymerase is used to catalyze DNA synthesis. So the primer binds to the DNA strand, and the nucleotides bind to the template in a random process. This means that either a normal nucleotide or a terminator nucleotide can bind to the template. And depending upon exactly where the terminator nucleotide binds to the DNA template, DNA synthesis may be terminated after a few nucleotides or only after a long fragment of DNA has been synthesized. So as a result, fragments of DNA in each tube will be of varying lengths. One thing they have in common is that all the fragments of the new DNA in any one tube will end with a nucleotide that's got the same base. For example, cytosine in tube 1 and guanine in tube 2. These fragments can then be identified because the primer attached to the other end of the DNA section is labeled radioactively or with a fluorescent dye. The DNA fragments are placed onto an agarose gel, and a voltage is applied across the gel. So the fragments will move through the gel towards the anode if they are negatively charged, or towards the cathode if they're positively charged. Now remember that DNA is negatively charged, so it's going to move towards the positive end. So due to the resistance of the gel, they move at different rates depending on their mass. So the details of gel electrophoresis are as follows. The gel is made and poured into a container. A comb is applied at one end to create small wells in the negative side. The gel is then left to set and the combs removed. Then you add the DNA fragments into the wells at the one end of the gel. So this is added to the negative side. You turn on the electrodes, and then because DNA is negative, the molecules are going to move towards the positive electrode. So the larger the fragments, the more slowly they move. And over a fixed period, smaller pieces of DNA will move further than larger ones. So in this way, fragments of DNA of different lengths are separated. Now, gels are colloids with spaces or pores through them, through which the molecules can move. And you've got polyacrylamide and agarose. So polyacrylamide gels have got small pore size, good resolving power, and they do separate small fragments, but they are quite difficult to prepare. Agarose is what you usually use for DNA. It's got good resolving power, large molecules, and it's quite easy to prepare. Now, the rate of migration of the DNA fragment is going to depend on a few factors. The size of the DNA fragment, the concentration of the gel, the voltage applied, and then the gel components and buffer solution. Now, your DNA is transparent, so you need to be able to view it somehow. So the first step is you need to immobilize and transfer this DNA from the gel. So this can be done by transferring the DNA fragments to a nylon membrane using the southern blotting technique. So here you can see how the DNA has been transferred onto a filter. And then you take this filter and you seal it into a bag with radioactive probes. So these radioactive probes are going to bind to complementary sequences. You can then identify your probes using X-ray. So using a gene probe, these are radioactive or stain probes of RNA or a single strand of DNA that's going to be complementary to the DNA strands that you need to detect. And this process might require quite a few probes. So if radioactive probes are used, then they can be seen by using X-ray film. Or you could stain your DNA fragments. So you could use ethidium bromide or methylene blue or Nile blue. Now, ethidium bromide fluoresces under UV light, ultraviolet light, but it is quite toxic. Methylene blue does fade quite quickly and it does stain both the gel and the DNA, as you can see here. Now, you've got your gel with the different sequences. How are you going to be able to sequence the DNA? Well, what you do is you read the gel from the bottom to the top. So this is because the shortest fragment moves the furthest. The smallest fragment will just be one nucleotide long and will therefore be the fragment near the bottom. So see if you can identify the sequence of this DNA from this gel. You can see that the first fragment is adenine, the next one is cytosine, then you've got thymine. So you're looking at where those fragments correspond to the different letters. Now, only DNA fragments of around 500 bases can be sequenced in this way. Larger genes and whole genomes must therefore be cut into smaller fragments. Do you remember what cuts DNA? Restriction endonucleases. So these will cut your DNA and then you sequence each fragment. DNA sequencing is now routinely done by automatic machines, and the data produced is going to be analyzed by computers. So in these computerized systems, instead of radioactively labeling the DNA primer, the 4 types of terminators are labeled with fluorescent dye. So each type uses a different color. For example, adenine is green, cytosine is blue, guanine is yellow, and thymine is red. So electrophoresis is carried out in a narrow capillary tube with the gel in it. The DNA results are scanned by lasers and interpreted in a fraction of time taken by conventional methods. Right, in summary, we looked at sequencing the nucleotides of a gene. So this involves making numerous copies of your gene by PCR, then using terminator nucleotides to identify the fragments. The fragments are separated using gel electrophoresis, and we discussed the agarose gel and the polyacrylamide gel. Agarose is usually used in DNA electrophoresis. The rate of migration depends on several factors such as the size of the fragment, the concentration of the gel, and the voltage applied. And then reading a DNA sequence from a gel, you start at the bottom because this is the first fragment and you go up the gel. And then we also looked at how DNA sequencing is now an automated process, which is much faster. And that concludes our lesson. The end.